Bismillahirrahmanirrahim. Assalamualaikum. My name is Amirul Faez bin Shamsudin, and the title for my oral presentation is Investigation of the Associations between Genetic Polymorphisms and Cholesterol-Lowering Effect of Statin among outpatient statin users in hospital USM. So let's move on to introduction. So what is statin? Statin is commonly used as medication to reduce cholesterol levels. And mechanism of action of statin takes place inside the liver during cholesterol biosynthesis process, in which statin inhibits conversion of HMG-CoA into mevalonate, therefore causing reduced production of cholesterol in the liver. So how does statin affect lipid profiles in plasma? Focusing on atherogenic lipoprotein, statin treatment able to reduce level of atherogenic lipoprotein due to increased uptake to the liver. While for non-atherogenic lipoprotein, statin treatment able to increase the level of HDL due to reduced transfer of cholesterol from HDL to atherogenic lipoprotein. Statin efficacy in reducing cholesterol may vary in different persons, and this varying efficacy may due to several factors which include genetic polymorphisms. This figure summarizes the gene that involved in statin disposition as well as statin efficacy and statin side effects. Now, talking about genetic polymorphisms, single nucleotide polymorphism is the most common type of genetic polymorphisms. And based on candidate gene approach, 8 SNPs within 7 genes have been selected for this association study, which is listed in the table below. Therefore, we hypothesize that these selected genetic polymorphisms are associated with cholesterol-lowering effect of statin, and our aim is to investigate the association between these selected genetic polymorphisms and cholesterol-lowering effect of statin among outpatient statin users in hospital USM. This slide summarizes the methods of our study. For first part, the patient's recruitment, a total number of 229 outpatient hyperlipidemic statin users were recruited from hospital, followed by DNA extraction and SNP genotyping. For SNP genotyping, a method called PCR-FLP was performed, in which the DNA extracted was amplified and then digested using restriction endonucleases. And finally, statistical analysis was performed using SPSS software, in which recessive model analysis was used to group the patients according to minor allele carriers and non-carrier group. And pairwise comparison between these two groups was performed using Mann-Whitney test and independent t-test. This table summarizes the PCR-RFLP protocol, including the primer sequences as well as restriction enzyme used for RFLP reaction for all SNPs. Moving on to the results, the first is patients' demographic profiles. Looking at the table, focusing on the bottommost part of the table, the mean lipid level at baseline, you can see that our population had elevated total cholesterol and LDL level above normal range. While for HDL level, our population had reduced as compared to the normal range, while for baseline triglyceride level was borderline as compared to the optimum level. The second result is the comparison of genotypic distribution of our population and a reference population using chi-square analysis. The reference population here is referring to East Asian populations from Ensemble database. As you can see in the table, focusing on the p-value of each SNPs, you can see that CETP RS708-272 and COQ RS4693075 was not significantly different as compared to reference population, while for the other SNPs were significantly different. While for ApoE genes, as you can see in the table, focusing on the p-value, you can see that genotypic distribution of ApoE gene was significantly different to the reference population. And our main finding for this current study, the first is ApoE5 RS662799. As you can see in the figures, that the minor allele carriers of this SNP had higher HDL level and reduced triglyceride level as compared to non-carrier after 7 to 12 months of statin treatment. Next SNP is ABCC2 RS717620, in which you can see in the figure that the minor allele carriers had a lower triglyceride as compared to non-carrier after 7 to 12 months of statin treatment. And finally, for ABCG2RS2231142, you can see that minor allele carriers had higher total cholesterol as compared to non-carrier after 7 to 12 months of statin treatment. For discussion part, for ApoE5, based on previous study, had been associated with decreased plasma triglyceride level that is caused by reduced VLDL synthesis in the liver. And you can see in the current study, the SNP of ApoE5 RS662799 carriers had a better response to statin treatment with regards to increased HDL and reduced triglyceride level. For ABCC2, RS717620, based on previous study, had been associated with reduced gene expression, therefore causing an increased statin bioavailability and enhanced efficacy. And our current study showed that these minor allele carriers for this SNP had a better response in terms of triglyceride reduction. And finally, ABCG2RS2231142 also had been associated with gene expression. However, our current study had shown contradicting findings, and we come up with one possible explanation that is due to drug class effect, in which based on previous study, ABCG2 had been shown to have preferences for certain statin. However, our study did not specify finding according to statin types, which therefore might give different findings as compared to previous study. As conclusion, statin efficacy is associated with genetic polymorphism, in which we found out that ABCC2 RS717620 and ApoE5 RS266279 had a better response, while for ABCG2 RS223114 had a reduced response. And for future study, we strongly recommend a future multi-centered study involving more SNPs with stricter recruitment criteria, in which will certainly help to improve understanding towards this field. That's all for my presentation. Here's a list of references, and thank you.